Hi there, my name is Grace Reynolds, and today we will be exploring the genetic differences between autoimmune disorders. So autoimmune disorders have previously been found to be comorbid and to share genetic loci. Currently, these similarities make it really difficult to differentiate between autoimmune disorders, particularly in a clinical setting. In hopes to better distinguish between them, we explore genetic differences amongst autoimmune disorders. So the main question we're grappling with here is what genetic differences exist between previously correlated autoimmune disorders? And the answer to this question should be able to lead to further insight that will help people all over the world receive better treatment and diagnosis of their autoimmune disorders. So here we perform case-case GWAS, also known as CCGWAS analysis with the tool REACT, using summary statistics data to explore the genetic differences between these cases of autoimmune disorders. This excludes the HLA region. The initial disorders we tested are ulcerative colitis, often shortened to UC, and Crohn's disease, often abbreviated to CRD or CD, and rheumatoid arthritis, shortened to RA, and systematic lupus erythematosus, shortened to SLE. This analysis resulted in many genetic loci that were then filtered against their original summary statistics. From these novel loci, novel genes were mapped. We also used the GSMR tool to find an association between our CCGWAS SNPs and tissue-specific gene expression, also known as EQTLs as the technical term. Here, whole blood was the tissue that we explored. Finally, we used another tool called Enricher to explore whether genes identified from the CCGWAS had significant overlap with the gene sets with genes that were already known to be involved in biological processes. The results of this gene expression test are known as enriched terms. Hopefully, this can lead to some information about the gene's role in the body. So the results we found from this process included a number of differing novel regions, also known as loci, and novel genes between the disorders that we compared. So our first comparison was made between UC or ulcerative colitis and CD or Crohn's disease. We found 35 loci, 4 of which were novel when compared to the original studies. And we found 6 novel genes that were located in 3 of the 4 novel loci. For our other comparison between rheumatoid arthritis and lupus, we found 65 loci, 38 were novel, and 21 novel genes that were found in 13 of the 38 novel loci. So we suspected to have more novel genes for the lupus and rheumatoid arthritis comparison because these two disorders are less correlated with a score of 0.451 than the ulcerative colitis and Crohn's disease with a score of 0.62. But this is only a suspicion about our results. So continued, the significant enriched terms for ulcerative colitis and CD comparison are mapped here below in figure 2. So as we can see, the circles in orange at the top are the gene names and the blue rectangles at the bottom are the terms. And both of the terms there are immune-related processes, which is quite promising. For the other comparison between rheumatoid arthritis and lupus, only one significant enriched term was found. That was negative regulation of T cell differentiation. This is still an immune-related process, though. So this is really promising and important for clinical use and for patients. There was no significant findings regarding the tissue-specific gene expression from earlier of the EQTL. So in conclusion, our analysis revealed differential loci between cases of correlated autoimmune disorders. Novel genes known to be important in immune-related processes were also identified. This study can be furthered by comparing more sets of autoimmune disorders to yield more results. In the future, these loci may provide insight for better diagnosis and disorder-specific treatment for autoimmune disorders. This is really important in a clinical setting. Thank you so much for listening, and I hope you learned.